in class, then you're going to come to class on Friday, ready for exam 4. No cheat sheet for exam 4, right? If you prefer not to take exam 4 next Friday, the 21st, then you're going to have to take exam 4 and exam 5 on December 10. That's our regular final day. Okay? Everybody's clear with that? Yeah. No. No cheat sheet on exam 4. No. You can have it on exam 5. Actually, I heard some students that said they never even had a cheat sheet for any of their classes. So just for fun. All right, so here, we stopped talking here about the mitochondria and how it is the business for mitochondria to shuffle energy. So let's focus. All right, let's focus. The doggy is a bit of an anesthetic distraction, not 100% distraction, right? So here, we are talking about fusion and fission of the mitochondria. Fission means, you don't have to guard the door. Okay, fission means it gets together. Fusion, see, it's so always confusing. Fusion is when it fuses together. The fission is when it divides, right? So here, why is that important for mitochondria to merge, right? So basically we see here that sometimes mitochondria can get damaged, especially the DNA inside of the mitochondria. It can be bombarded with free radicals because a lot of things are happening in the mitochondria, especially when you're going to use oxygen, right? When you're going to get to your oxidative phosphorylation type of energy production, right? So you're going to have free radicals running around. You have a lot of electrons that are carried around inside of the mitochondria. Some of those electrons can go rogue and decide to hit the DNA and do DNA damage. When DNA damage happens in the mitochondrial DNA, that's a big problem because mitochondria do not have DNA repair mechanism, right? Just like with bacteria. Bacteria don't really have DNA repair mechanisms. Right? Well, some of them do, but mitochondria doesn't have it, okay? So then what is going to happen? When you have some damage of the mitochondrial DNA, you have two choices for the mitochondria. Either the mitochondria can be discarded, like in mitophagy, right? Or you can have fusion of two neighboring mitochondria, and then the mitochondria that has the good DNA can share the DNA with the mitochondria that had the damaged DNA. Okay? How many copies of DNA does each mitochondria have? A lot. Up to like out of 5,000 or so of those circular DNAs, right? Remember that besides circular mitochondrial DNA, you could also have some plasmids. Okay. So here, what happens here? If you have fusion of two mitochondria, you can donate some good DNA to the mitochondria that has a bad DNA, right? And then you can salvage that mitochondria. Also, another thing is of this fusion and fission business is that you need to keep up with how much energy your cell needs. Okay, we're going to get into a little bit more into that story now. And as I promised you, I was going to put some more iClicker points here because we're missing it from last time. Just a second. All right, so here was from the last class. The question is, what is the end product of glycolysis under aerobic conditions? Now, this is specific, okay? In a rapidly contracting muscle cell, okay? And what do you need? You need to regenerate NAD plus in that muscle cell. Right? So what is the end product of glycolysis? Now you need to remember that glycolysis is happening in the cytoplasm, right? Now, we have only one correct answer here. You want to regenerate your NAD+. So some people who are confused are giving me the right answer. Some people are still confused. Oh, okay, no, this is going the wrong way. Some people are getting more confused. They had the right answer, now they're giving me the wrong answer. 5 more seconds. Pick one. Need to regenerate your NAD+. 20 seconds. That's how long ChatGPT is going to take to give you the answer. Oh, we got a 50-50 split here. Okay, I'm going to stop it. Did everybody at least get 1 point for participation? Yes. Okay, so I'm going to stop it. What is your answer? YMC. Hey, yeah, this is what I put the specifics in the question, okay? You are undergoing a lot of muscle contraction, very fast muscle contraction. You want to recycle your NAD+, because that's the only way you can keep glycolysis going, right? So you are going to dump those electrons from the form of pyruvate to make lactic acid. So lactic acid is correct answer here. Yeah. What? Pyruvate, yeah, is the end product of a glycolysis, but if you don't need NAD+, right away, you're going to see more on pyruvate. Remember fates of pyruvate? Yeah, I know this topic is a bit confusing, and it takes a lot of brain power. Yeah, that's why I brought Lola with me today. My psychiatric service dog. All right, so let's look into more details in mechanisms and things that are important for energy generation, okay? So we are looking here before at what happens when two mitochondria are going to fuse, okay? So now what is going to happen here? You're going to have first thing, the outer membrane of the mitochondria is going to fuse because they're going to come in contact first, right? Remember, mitochondria has two membranes. So when the outer membrane gets together, you are going to have proteins that are going to use some energy. Now, if you think about it, if something is happening here with the mitochondria in general, you are really trying to salvage mitochondria to produce ATP energy. So when you're going to do some process here with this mitochondria, you don't want to use up more ATP because you're already in a deficit. So the energy that you're going to use here is the energy generated from GTP, okay? So GTP energy, you're going to have some GTPases that are important proteins and enzymes on the outer membrane, okay? So we're going to have this outer membrane GTPase that is going to help to fuse the outer membrane. That's simple. Once the outer membrane is fused, then you're going to have fusion of your inner membrane, okay? So to conclude here the fusion of the outer membrane, you have your GTPases, and you're also going to have specific proteins that are all specific of those GTPases that are called, at least this guy is easy, okay? It's called mitofusins. So the name of the enzyme is telling you what it's doing, right? Mitofusing is going to join this outer membrane over there, okay? So then once you have your outer membrane fused, now we're going to deal with your inner membrane. Here's the fusion of the inner membrane. You're also going to need GTP, okay? And you're going to have those enzymes here. They're going to be responsible for fusing that inner membrane. Now those enzymes are a bit more complex than the enzymes that are fusing the outer membrane, because now you're going to have to deal with an environment here that is highly acidic, It has a lot of protons over there. You have your proton gradient between the membranes, right? And you have to conserve the environment inside of the matrix, right? So those enzymes here, they have more work to do. They have to be more careful than the enzymes that fuse the outer membrane. So one enzyme that is important for you to remember is this guy over here, and it's color-coded, okay? Optic atrophy 1. Yeah, it does not tell you inner membrane fusing. I wish it would, but no. It's called optic atrophy 1, and is abbreviated OPA1, okay? So OPA1 is going to use GTP, is going to fuse the inner membrane, and then you're going to have this bigger mitochondria here that is going to share, some mitochondrial DNA and some other factors that maybe one of those mitochondria were in need. Okay. Now, why is it called optic atrophy 1? Because there is an inherited disease in which this protein is not working properly. And when mitochondria do not work properly, some cells, especially special sensory cells, meaning in your eyes, that need a lot of handling of energy, they do not work properly. And you start to have retinal degeneration, or you're going to start losing the vision. And this is the protein that is responsible for that. Okay? So optic atrophy 1. There are other proteins important for that as well. If we have time, I'll tell you a bit of a story on mitochondrial inheritance. Right? By the way, how do you inherit your mitochondria? From the mother. It's maternal inheritance. Okay. So here it is, right? So you know that your genome is made out of 2 things. You have your linear DNA that is in the nucleus, and you have your mitochondrial DNA that is, of course, in the mitochondria. What do you find in the mitochondrial DNA? You're going to find not a lot of genes. Remember, it's circular. You're going to have 37 genes. Now look at this. You're going to have a lot of copies of the genes per mitochondria. Okay, a lot of them. Average 5,000, right? Now, this is an observation that I put here. Because of the time, what is the matter with you? We're not going right now. We're going to sit down and behave. She doesn't want to learn about mitochondria. Okay. So what is going to happen here? The older your mitochondria get, the more the DNA is going to be exposed to reactant oxygen species, to free radicals, right? And the more damage you're going to get in the DNA, right? So the older the mitochondria gets, the more damage you're going to have in the DNA, okay? Now, this goes without saying that aging, cellular aging, and then subsequently overall aging, has to do with the state of mitochondrial health, okay? She doesn't like cell bio. All right. So here, looking at the genes that are present in the mitochondrial DNA, okay, you have 37 genes. 13 of those are involved with producing enzymes that are important for electron transport chain. The rest of the genes are important for production of transfer RNAs and ribosomal RNAs. And now let's think about it. You have here your circular DNA in the matrix. So here, you are going to produce tRNA, ribosomal RNA, and you're also going to produce messenger RNA to make those proteins that are important for your electron transport chain. Okay? When is it that you're going to activate your mitochondrial DNA to make those proteins? Well, basically, when you need to produce more energy. Or let's say when some of these complexes here are kind of like failing, and you need to, replace a piece of the complex. So then you're going to activate your DNA, you're going to make ribosomal RNA, tRNA. Those are made constantly, right, in a housekeeping type of expression. But then you're going to upregulate those messenger RNAs. You're going to make the protein, and then the protein is right there to be placed at the complex where it's needed. Okay. Now, this whole complex of electron transport chain uses over 70 different proteins. And then mitochondria is producing only 13 of those. Where is the rest of the proteins coming from? Guess, take a guess. Okay, you just have to use your brain for this one. Mitochondrial usage of brain power. How people think? Yes? Oh, which one? Exactly. It's coming from somewhere else, like your nuclear DNA inside the nucleus. Do not run after this job. Sorry. So, remember Tom Tim? Remember Tom Tim, Tom Tim, outer membrane, Tim, right? Tom. Those Tom Tim guys are going to import proteins from the cytoplasm. Remember protein import? Yeah, protein is important for mitochondrial function. So here, let's think. Now this is important part, okay? Let's focus here. Not on Lola and the bear. Important thing that is happening here. This is a concept that is not explicitly written, but you need to understand. Everybody focus? Yeah. Why not have all of the genes coming from the cellular linear DNA? Why do you need to produce locally some of these proteins? You don't need to try for it. Question is, why to produce proteins locally in mitochondria instead of bringing everything from the linear DNA that is inside of your nucleus? Huh? They could come that way. They could come. You have protein transport. You could bring proteins from the nucleus that originated a messenger RNA in the nucleus. You could bring them, but why not? Why does mitochondria keep some of those genes? Yeah. That was a guess saying maybe because it takes more energy, but no, that was a good guess though. Yeah.
Maybe because it's for DNA, so
No, you're thinking about immune system defense. For DNA, no, not really. Yeah. Yes, this is one of the reasons. It is faster. Okay, that was disaster. It is faster. So we want to have local production. You want to have local production, right? But the reason is because you want to have the local production at that mitochondria. You are distracting the class. Relax here. Local production. Why? Let's say one mitochondria is defective. You need to substitute this piece over here. And you're going to wait for protein to be produced in the cytoplasm. Go to the translation, and then remember that protein must have a signal peptide sequence is going to be stretched out with GTP energy. It's going to be pushed through TOM-TIM, right? It's going to get to mitochondria. Okay, now, which mitochondria? Let's say you're producing the protein in the DNA, in the nucleus. Then you're going to send a signal, a sequence for the mitochondria. This one mitochondria is on the corner of your cell, and it's saying, I need it. And how do you know to send the protein to that mitochondria? You just have a zip code. So zip code for mitochondria. You have about 10,000 mitochondria in your cells. So now you have a problem. An analogy is that you want to send a letter to one of your friends that lives in New York City, and you only know the zip code. Good luck, right? How is your friend going to get the letter? Well, you better send a lot of letters that hopefully one of them is going to reach the right address. But that's a big problem, okay? If the mitochondria has to wait for the protein to be produced, and then the protein figure out, it's going to go to all the mitochondria, maybe this one is going to get it, maybe it's not going to get it. By that time, mitochondria is dead. Okay, so you need to have some local production of essential genes that are part of your electron transport chain. So today is going to be everything about electron transport chain. All right. So here, recap what is happening in the mitochondria with more details. When you look at the mitochondria outer membrane, you know that this outer membrane has to be more permeable than your inner membrane. Okay? So here in your outer membrane, you're going to have certain channels that are called porins, okay? Certain little channels over there. And these porins are going to allow certain things to go through the outer membrane without a lot of checkpoint, okay? So small things, like you look at this, outer membrane is permeable to ions, right? Also to some metabolites and some little molecules, up to 5 kilodaltons, small molecules. And importantly, it is permeable to ATP, ADP, and phosphate. That's crucial, okay? So ATP, ADP, and phosphate can go through those porins and reach this inner membrane space. Okay. When you look at large proteins, those have to go through the transporter of the outer membrane, through TOM. Okay, but little things can go through these little pores. Okay. Okay. Now the inner membrane is a different story. Okay, so the inner membrane is very much impermeable to anything, right? Especially to ions. Because if the inner membrane gets permeable to ions, you're going to lose your gradient, and then you cannot make ATP, right? The electron transport chain is going to happen at the inner membrane. So that one is highly impermeable. You need very controlled transport through your inner membrane. All right. So here, at the inner membrane, you're going to have these translocases, the ANTs, okay? So this is the ATP, ADP translocase. What is going to happen? In one side, you're going to get ATP, it's going to bind to it, it's going to dump to the other side. You're going to get ADP, it's going to bind to it, it's going to dump to the inside, okay? So ATP and ADP need a translocase. Then you're going to have another translocase here that is going to transport phosphates. Why do you need phosphates in the matrix? To put it on ADP and make your ATP. Without the phosphate, you cannot make your ATP. Okay? So you have these very important transporters in the inner membrane. Yeah. No, it's not active. Because if you need energy to make those transports, you're going to be wasting the energy the mitochondria is making. So yeah, so those are just transporters. Specific transporters, but they're just facilitated diffusion. That's the proper term for it. Okay? What else are you going to have in the inner membrane? You're going to have a calcium transporter. That happens to be unipolar. You have the GAI-TIM, right? Oh, and then you're going to have this pyruvate transporter. That's going to be very, very important, right? And you're going to have this mitochondrial permeability transition pore. Okay, that is not your friend. Why? When you open up this guy, this is when the cell as a whole decided to commit suicide. Okay? So if you open this guy, the mitochondrial permeability transition pore, MPTP, when you open that guy, you're going to let cytochrome C out. Okay? And that means cell is going to die by apoptosis. All right? So this guy here, let's keep him quiet, unless you really need cell to die. Okay? All right, so here overall picture of the mitochondria. So you can read these summaries on your own. This is from your book. Okay, let's get the fun stuff. I just finished reading this book, which is really a very cool book. I took that quote from Star Wars out of this book. Okay? So if you are going to healthcare professions, I would advise you to, I didn't read it, I just listened to it, audiobook, very practical. Mitochondria and the future of medicine. So here, back to energy production, everything about energy today, right? You are going to need to get the pyruvate from the cytoplasm inside of your mitochondria, right? Because you're going to have to start your citric acid cycle, you're going to generate your NAD, FADH2, and you're going to pump those electron transport genes. So you need to get pyruvate in the matrix, okay? It's not an easy task. From the cytosol, where are you going to get pyruvate from again? Glycolysis. Where is glycolysis happening? In the cytosol. Is that a test question? Yes, that's the clue. Okay? Don't tell me I didn't tell you. Okay? All right. So here, you get your pyruvate. When it comes through the outer membrane, look at this, it's going to diffuse through those pores, no problem. Okay. When it's going to get to the inner membrane, that's when you're going to have this transporting business going on over here. Okay. You're going to need this mitochondrial pyruvate carrier type of thing. Okay. So here you're going to have this co-transport of pyruvate that is going to go with one proton and is going to be helped here to get inside of the matrix. Okay. Now in my previous slide, in previous year, I had put here secondary active transport, but that is actually not right. The correct idea for here, it's working more like a facilitated diffusion type of transport. But then you should argue now saying that, how did you get the hydrogen concentration over here? Well, you had to use some energy of electrons or something. Yeah, I know, there is a gradient. You need energy for a gradient. Then this guy is going to go down, energy gradient. But what makes this different from secondary active transport is that the pyruvate that is coming with the hydrogen is also going down the concentration gradient. You're not pushing anything against the concentration gradient. Everybody got that? This is a detail that sometimes people miss. Okay? You are not pushing pyruvate against the concentration gradient. It's just allowing it to diffuse to the matrix. Okay? So this is more like facilitated diffusion. Okay. Okay? So we got the you got the pyruvate there. Once pyruvate gets inside of the matrix, it is going to be oxidized. You're going to have this huge enzymatic complex that you call the pyruvate dehydrogenase complex. It's a monster. It has at least 3 arms. Okay? So here is a little video of how that monster works. Okay? So first thing you're going to get, here's pyruvate. You're going to get to the little pore. This is happening, okay, the mitochondria. Okay, this is from the outside. You go through the pore. And now here you're going to go to the inner membrane. And now it's going to hit the matrix. And there's the monster. You see? Scary monster. All right. That's your pyruvate dehydrogenase complex. Okay? What is happening over here? Pay attention, right? What is happening over here? You're going to have at least 3 enzymes. They are going to work here. Okay? The first one is your dehydrogenase. You're going to get pyruvate. You're going to cut a piece of it as carbon dioxide. And you're going to have acetyl left. Then you're going to have this enzyme 2 that is going to be transacetylase. Okay? What is that going to do? Here is your enzyme. Is going to get CoA and it's going to put in acetyl to make acetyl-CoA. There is your acetyl-CoA, right? And then you're going to have your pyruvate, oxidized pyruvate over here. Let's see here what else is going to happen. Third enzyme, also dehydrogenase. You are going to get the energy that came out of the oxidation of pyruvate, and you're going to make NADH. Where is NADH? It's right there, the little light green one. NADH reduced, right there, see? Right there. So what happened over here? You started with pyruvate. You cut carbon dioxide out of it. You're oxidizing pyruvate, so something else has to be reduced, okay? Then you're going to get the energy and put hydrogen on NAD and make NADH. Okay. Details on that. So here, pyruvate, right there. So first thing that happens, carbon dioxide gets cut out. 2 pyruvates because you always start with glucose, right? Here you're going to get your NADH, and then you're going to have molecule of CoA, and you're going to end up with acetyl-CoA. Everybody cool with that? What is happening? Pyruvates got oxidized, and then your NAD plus gets reduced to NADH. Hello, what's up in this class? Yeah. So here, your acetyl-CoA, right? Acetyl-CoA is a very, very important molecule. It's actually the center of metabolism. That means it's the end product of catabolic reactions and is the start product. Okay, guys, let's be quiet. Okay, is the start product of anabolic reactions. So what kind of catabolism? Let's see here, because you're going to have another question coming up, right? If you're going to break down fatty acids, okay, you're going to end up with acetyl-CoA, 2 carbons. If you're going to break down starch, glycogen, sugars, you are going to end up with glucose, and then you end up with pyruvate and acetyl-CoA. If you're breaking down amino acids, you're also going to end up with acetyl-CoA. So you can break down proteins, fats, and sugars. Those are all of your macronutrients, right? Anything else other than that that you can use for energy? There is one more thing that you probably need after this class. All right, let's use your imaginations. Alcohol, exactly. You could use some alcohol here. It's not in the pathway because alcohol is actually toxic. It's not a nutrient, right? But you can use for energy sometimes. All right. So you can break down all of your macronutrients, get acetyl-CoA, and then you can use it to build up all of those things again. Look at this. You're going to use acetyl-CoA to build up cholesterol, biosalts, vitamins like vitamin K, okay? Fatty acids. This is a very important building block for anything else you're going to make in the cell. So here, you're going to have conversion of catabolism, diversion into anabolism, and then you can use that to produce your energy in the mitochondria. Okay? Now, the acetyl-CoA is this very, very important intermediate and is part of anaerobic respiration because you do not directly need oxygen for your citric acid cycle to take place. Okay? Or until here, you do not really need oxygen. Part of anaerobic respiration. Okay. So now let's see what happens here. How else can you get here? The question of your acetyl-CoA, right? So what is the balance here of when you're going to produce your acetyl-CoA, right? So here's the idea that says, let's say that you're kind of hangry. Everybody heard this term before? Hangry. Yeah. Hangry is angry and hungry, right? So if you are hungry, you're producing some hormones like glucagon, right? And then if you're angry, you're producing stress hormones like cortisol, maybe some epinephrine. Okay. So those hormones are going to increase the breakdown of fats, okay, in your body. So what is going to happen here? You're going to use fat to produce your acetyl-CoA, sometimes also protein, right? You're going to break down whatever you have in your body already available to produce your acetyl-CoA. Okay. Now let's say that you are not hungry. Let's say that you just had a nice meal and you are satisfied. You're in your parasympathetic state of rest and digest. Right? So what's going to happen? You're going to produce other hormones like insulin, right? Insulin is going to help you to pick up glucose from interstitial fluid, right? And then you're going to use the glucose that you just picked up, sugars that you just ingested, for breaking down into acetyl-CoA, and then you're going to use that for energy. Okay? So where you can get it? You can get from proteins, from fats, or an alternate example here is really what I mentioned, like alcohol is not a macronutrient, but it could also be converted in the liver to acetyl-CoA. All right. So here, let's see what else is happening here with your mitochondria, right? So acetyl-CoA is being produced in the mitochondria. Remember the monster of dehydrogenase, right? And you are going to end up with those electron carriers that are charged, and those are going to be sent to your electron transport chain. Okay? So here, another question. What is this image showing you? This is an easy question with scary words. All right, people, do you see any phosphate around here? No, so don't tell me D. Does anyone need more time? Yes? So 5 seconds. Everybody voted? Okay, what is your answer? B. Okay. This guy here is your pyruvate oxidation. So now let's get into the Krebs cycle, because they're very, very important to generate your charged electron carriers, right? Who took biochemistry? Or who is taking biochemistry? Remember you had to memorize all the stuff in the Krebs cycle? No? You didn't have to memorize this in biochemistry? What is happening nowadays? I had to, and I already forgot it. Maybe that's why you don't. Oh, okay. So who took the Krebs cycle in the whatever part of biochemistry that teaches it? Nobody? Oh, one person. Yeah, you had to memorize this? Yeah. So I'm glad everybody else has to go through the torture. It was not only me. Okay. So what is happening here? Overall idea for when you take it, you have to take biochemistry, right? You're going to have 8 enzymes that are important for your citric acid cycle. Okay, it's also called tricarboxylic acid cycle, TCA. Okay, you're gonna have enzymes here. Here we're not showing all of them in details, but basically look at this. 4 of those enzymes are called dehydrogenases. So half of your total enzymes in the TCA, they are dehydrogenases. What are dehydrogenases going to do? They are going to charge your electron carriers. So look at this. First dehydrogenase is going to charge NAD+. You're going to get NADH. Second one, more NADH. Third one, you're going to get FADH2. And the fourth one, you're going to get more NADH. Okay? So this is all happening because of your acetyl-CoA molecule. Right? So you end up with 4 charged electron carriers out of your... Okay. You see this guy right there? Enzyme number 4, that's what we're focusing on here. When you are getting this intermediate that is called alpha-ketoglutarate, and you're going to make succinyl-CoA out of it, you have this enzyme complex right there, alpha-ketoglutarate dehydrogenase. That is a very, very important complex for the TCA. That's the one you should know. Okay? All right, so here we go. Take a deep breath. You need it. Put more blood into your brain, like, yeah, it takes brain power. Okay, so here, alpha-ketoglutarate dehydrogenase complex. You're going to get this molecule of alpha-ketoglutarate, and you're going to need a lot of cofactors. Those are vitamins in order for this thing to work. You're going to need thiamine. That's your vitamin B1. You're going to need lipoic acid. That is an essential fatty acid. You're going to need coenzyme A. That is also your vitamin B5. It's also called pantothenic acid. Okay? You're going to need FAD. That's vitamin B2. Also known as riboflavin. You're going to need NAD. So FAD, riboflavin, NAD is your B3. That is your niacin. Some mnemonics for you to remember some of these. NAD, niacin starts with N. Okay, that's B3. FAD, riboflavin has F in it. Okay, and the rest we have other things to make you remember. And this is when you have these T-Rex guys, right? And they're saying here, T-Rex loves and cares for nachos. That's what should make you remember all the cofactors that are needed for your dehydrogenase complex, alpha-ketoglutarate, right? So Tregs loves and cares for nachos, okay? If you have deficiency of any of those vitamins, you're gonna be in trouble. So since this is not medical class and you're not focusing on diseases, but then if we don't, it becomes very boring. So I put here a couple of examples of diseases that are associated with lack of a couple of those vitamins. Okay, so the first one is beriberi. That is going to happen if you lack vitamin B1. Then you're going to have another one that's called pellagra. That is lack of vitamin B3. Okay, so niacin. So let's look at them over here. So yeah, you have to take those. If you don't take them, you all right. So here, clinical correlation. If you have lack of vitamin B1, you're going to develop beriberi. Okay. What happens with beriberi? Your heart can't work properly. Your nervous system can't work properly. Okay. If you have lack of niacin, you're going to develop pellagra, and the characteristics of pellagra can actually be reminded by 4 Ds. That is diarrhea, dermatitis, dementia, and death. Okay, serious stuff. So here in more details. First one here, beriberi, right? Thiamine deficiency. What you're going to feel if you do not have thiamine in your system? You're going to feel weak. You're going to feel tired. You're going to have a lot of pain. You're not going to feel your hands and your feet. You're going to have ataxia or difficulty walking. And in more severe cases, you're going to be paralyzed. Okay? You're going to be very confused throughout the process. Then your heart is going to start failing. And dementia or dementia with the mental confusion, and you're going to start getting very swollen because of course your liquid, your blood is not flowing properly. So those are all clinical correlations of beriberi. We do not see in this country or first world countries because most of foods are a lot of times are supplemented with iron. So the other one here, pellagra, very ugly, okay, pellagra, niacin deficiency. You're going to see a lot of skin rash. Another sign is what they call a strawberry tongue. Okay, I don't think I put it there. But yeah, it's like smooth and swollen and very red tongue, okay? And you're going to have problems with, diarrhea, besides the dermatitis, and then dementia and death. Okay. Do those make like good test questions? Because everybody remembers the ugly stuff that happens with the clinical correlations, right? All right, so here, back to energy flow. What you need to know out of this thing here. Okay. So you need to make sure that you get electron flow. It is electricity. Everything in your body is working because of electrical current. Your brain is working because of electrical current. You can hear what I'm saying because you can transform the sound waves into electrical currents. And then you can depolarize some neurons that can interpret the sound. You can see because of electrical current. You can taste food and smell stuff. Everything is electrical current, including production of energy. Right? So we have to talk about how is it that you get the electrical current to produce your energy. Okay? You have to work and look at the reducing ability of elements that are involved in this electrical current. Because when you think about properties of elements, like periodic table, property of elements, you're going to have electronegativity. Everybody remembers electronegativity? In basic chemistry, in the periodic table? Like electronegativity goes this way. It goes up. Like I'm going to your, like it goes to your right and to the top, right? So you're going to have a very, very highly electronegative element if you, oh, there's a periodic table, that's convenient. Look at that. It goes to the right and to the top. Which one is your most electronegative element? Yay, that's the F. All right. So now let's see what is happening over here and how you're going to make that into a useful information. Okay. So here, you are going to create electricity by carrying those electrons, using those electron carriers from your TCA. And you're also going to need the electron carriers from glycolysis. Glycolysis that happen in the cytoplasm. So remember those charged electron carriers over here? They're going to have to come into the mitochondrial matrix. You're going to have to put it there. Stop crying, it's not that painful. Okay, so here, how are you going to get it there? This is one example of how you're going to get your NADH into your matrix. It's not just going to flow through a channel. There is no channel for NADH, which is a good thing. Okay? So you're going to have to use a trick. You're going to have to use a trick. Look at this. In this cytosol, you are going to have an enzyme that is going to steal the hydrogen with the electrons from NADH and recover your NADH plus. This is in the cytosol, okay? So this enzyme is going to get oxaloacetate in the cytosol and is going to transform it into malate. You're going to recover your NAD plus. Now malate has a transporter. So malate is going to come from the outer membrane into the inner membrane, is going to get in the matrix. When malate is in the matrix, you're going to steal the electrons that it is carrying and transform it again into oxaloacetate. Okay? Now you're going to recover here your NADH with your 2 electrons. And then oxaloacetate is going to be isomerized into aspartate. And then you're going to have a transporter for the aspartate. So you call this shuttle the malate aspartate shuttle. Okay? And I have more picture of that. Here's a better picture of that. Okay? So in this cytosol, you are going to have oxaloacetate that is going to go through this enzyme that is called malate dehydrogenase. Okay? Now this is a bit tricky because you have 2 forms of malate dehydrogenase. One of them lives in the cytoplasm, another one lives in the mitochondria. Those 2 are not the same. They are not interchangeable. The one that is in the cytoplasm lives there. It doesn't go inside the matrix. Okay? So here, you get your malate dehydrogenase. Then you get your malate, you push it in. And now you recover your NADH, and then you transform this into aspartate. You push aspartate out. To recover your oxaloacetate, you're going to use this enzyme that is called aspartate aminotransferase. Again, you're going to have 2 isoforms for that enzyme. Okay? So this is a very, very clever shuttle of electrons into your matrix. Okay? Besides that, you have certain cells in your body that require a lot more energy and require fast and efficient shuttle of electrons. And those cells are part of your brain and also your muscle cells. Okay? So what's going to happen here? You have this additional shuttle that you call glycerol 3-phosphate shuttle. Okay, so this is especially for cells that require a lot of energy at once, right? And hopefully your brains are using a lot of this shuttle right now, right? So what's going to happen here? In the cytoplasm, you're going to have this dihydroxyacetone phosphate that is going to be converted to glycerol 3-phosphate. You're going to use the enzyme for that. The enzyme is glycerol 3-phosphate dehydrogenase. Okay, so here you are going to get NAD plus. You're going to get NAD+ where? In your cytoplasm. That means you loaded your carrier molecule, you made glycerol triphosphate out of it. This molecule is charged. It is going to be imported, okay? When it gets to the matrix, you are going to use the enzyme that is glycerol triphosphate dehydrogenase in the matrix, and you are going to take out the electrons. You're going to put it now into FAD to make FADH2, okay? Then you're going to recover your transporter. That's going to be outside again. You know, your transporting molecule going to be pushed outside, and this cycle is going to generate some FADH2. Okay. Your other cycle, malate aspartate, gives you NADH, right? And this one here is good for high energy, high demanding cells like brain and muscle. All right, let's make some ATP. All right. So here, how do you make the ATP? You're going to have to go through the electron transport chain. The chain is made up of several proteins that are embedded on the inner mitochondrial membrane. Some of those span the entire membrane, and some other complexes are only in the inner part of the membrane. Okay, so the three complexes that pump hydrogen into the inner membrane space, those are complex 1, complex 3, and complex 4. Okay. Complex 2 here is not going to pump hydrogens and electrons. Okay. So how is this going to happen? First, you're going to get your charged electron carrier. So with complex 1, you're going to get an NADH that is going to get in there and dump this high energy electron into complex 1. So electron here is very, very high energy. Then complex 1 is going to get this high energy thing, like remember the hot potato game, right? It's going to get this high energy thing. It's like, I don't want to keep this high energy thing. I want to push it into something else. So I'm going to, how am I going to dump it into something else? I'm going to throw it over there. And when I'm throwing it over there, I'm going to turn around. And with the turning around, you're going to get hydrogen that gets pumped into their inner membrane space. And turning around pushes the electron into something else that is going to pick up that electron. Okay? Now, this electron came from an ADH from complex 1. By the same token, you have the FADH2 that can dump this high energy, but this goes into complex 2. Complex 2 does not span the membrane. Complex 2 is going to dump these electrons into the same carrier that is coming from complex 1. But complex 2 is just going to free up FAD for it to go and pick up more electrons. Okay? So then this carrier over here is going to go to complex 3 and it's going to dump electrons over there. Complex 3 does not want to hold the hot potato. What is it going to do? I'm going to throw it somewhere that has more electronegativity than I have. So I'm going to do the turning and throwing. It's going to shift it to another electron carrier, and this guy is cytochrome C. Okay? This guy here is ubiquinone. This guy is called cytochrome C. And then cytochrome C is going to pick up and throw it to complex 4. It doesn't want to keep the hot potato, right? So it transfers to complex 4, and then complex 4 is with the hot potato, and it's going to say, I don't want the hot potato. I want to shift it to somebody else. And what's it going to do? Throws the electron to where? Oxygen. Oxygen, look at the periodic table, is very close to fluorine or fluoride, never know in English. The F, okay? Oxygen is very close to the F over there. It's highly electronegative. So here, you are going to use oxygen and make water out of it, right? So here you are going to oxidize. You're going to reduce the oxygen to become water, okay? And at the same time, you're going to pump another hydrogen into the intermembrane space, okay? All right, so here's what is happening, right? Important things to know here. Where do you dump your electrons from NADH? You're going to dump it into complex 1. Where do you dump electrons from FADH2? You're going to dump it over here in complex 2. Then you're going to shift this into ubiquinone. The electrons are going to pick up on ubiquinone. Ubiquinone is very, very important. Actually, for proper function of ubiquinone, you need a cofactor that is called CoQ10. So you know health food stores, you can buy CoQ10? Yeah, it's good for ubiquinone electron transport. It's a cofactor, okay? So now what's going to happen? Ubiquinone is going to transfer electron to here, complex 3, but it could also, it could also transfer electron to cytochrome C that goes to complex 4. So ubiquinone is a very, very important traffic controller of your electrons, okay? So here, let's see here what happens. When is it you're going to have your pump? 1, 3, and 4. Yeah, she's not a happy camper. All right. So now let's see here what is going to happen, okay? Again, here just to reiterate where you dump those electrons, okay? And here you're having your CoQ that is going to pick up those electrons. So this is just in a form that you can study, okay? Now it gets interesting. For this ubiquinone transporter, you're going to have two forms. You can have ubiquinone, or you can have the reduced form that is ubiquinol. We're going to have a picture for that. I love this slide. All right. So if you have oxidized form, you're going to have a dione right here, dione, okay? Selene dione, okay? So oxidized form is a dione. If you put electrons on it, you're going to get the alcohol forms. So you're going to have diol forms. So there you have selene diol. Okay? Dione, diol. You can carry 2 electrons, and of course they always go with the hydrogens. Okay? How is it going to shift those electrons? It is going to donate 1 electron at a time. Okay, so you're going to have formation of free radicals for shifting of those electrons by ubiquinone. Okay, ubiquinone. All right. So this guy here, your CoQ, is a very, very important traffic cop that is going to pick up things from complex 1 and complex 2, and is going to shift that into complex 3 and also into cytochrome C that is going to get to your complex 4. Okay, I know it's a lot of information here. All right. So here, what is going to happen here? This slide here, don't panic. This is FYI. Okay? In case you're interested to know the mechanism of how that is happening, right? So you have different forms of your CoQ that can be carrying 1 electron or 2 electrons, and then how those are going to deliver to complex 3 and complex 4. So why do you need to know this? You need to know that your CoQ is going to deliver electrons to complex 3 and complex 4. That's all. All right. Okay. Now, fun part, toxins. You have some poisons that can stop your electron transport chain. You have some of them that do not kill you right away. You might survive. Example of it is rotenone. Have anyone heard of rotenone? No? Is rat poison. Okay, so rotenone. What this thing is going to do is going to block complex 1 right there. Okay, so rotenone is going to block your complex 1. if you block complex 1, can you have a proton gradient in your inner membrane space? Okay, you gotta use your brains now. Everybody awake? Yeah? Who is not awake? Yeah, of course not gonna answer. Let's go for votes here. You gotta vote, okay? How many people say yes, you can have a proton gradient in your inner membrane space? You gotta vote. Go for it. Go for it. You got 50% chance of getting it right. If I hand my hand up, that means yeah, you can have it. So who's gonna vote yes? Should be everybody. How many people think no? Hopefully nobody. Okay, how can you get a proton gradient if your complex 1 is not working? You got complex 2 working. You got complex 3 working. Complex 2 can get electrons from FADH2. So now you can still have some pumping of electrons by using complex 3 and then using complex 4. So this thing is not as deadly as it could have been. Okay. Then you have another thing here, antimycin A. This guy blocks complex 3. Now you're pretty much doomed, right? Although you can bypass complex 3 by putting electrons on CoQ, and then this guy is going to shift to here. You still get a little bit of protons by complex 4. So it's more deadly than rotenone, but it's not as deadly as it could be. But now we get to the very deadly ones, okay? Cyanide. I'm sure everybody heard of cyanide. Carbon monoxide. You heard of that one? Carbon monoxide. And also sodium azide. Those guys can block complex 4. Now you're dead. Dead. Bones over there, right? Why? Because now there is no way that you can pump electrons into your inner membrane space. Cyanide will block there, so it blocks the transfer of electrons to oxygen. Right? Carbon monoxide blocks your complex 4, and actually I like to tell you here, carbon monoxide is very good at killing you because it kills you twice. The first time is by blocking complex 4. Second time is by kicking out oxygen from your hemoglobin molecule. Okay? Carbon monoxide will take the place of oxygen and get stuck in iron, and then you cannot transport oxygen to your cells. Very, very deadly. Okay? All right. And then there are other things that you can do to your inner mitochondrial membrane to mess it up, right? There are these things called uncoupling agents. Okay, so uncoupling agents. Have you heard of DNP, TNT? No? What is TNT? Some explosive of some type? Yeah, you make dynamite with it? Yeah? So look what happens here. These uncoupling agents, look at the shape of this thing. It is very hydrophobic. It has some polar sides, but this thing can go to the inner mitochondrial membrane and put holes in it. So then you're going to lose your electron, your proton gradient. But when it goes through the membrane, you are going to produce heat. So then you're producing heat, you're overheating your mitochondria, and you're not getting energy, you're not getting ATP, because your gradient is broken. Then you have to consume, you have to break down a lot more energy sources, macromolecules, in order to keep your energy production. And what is that going to do to you as a whole body? It's going to increase your metabolism. It's going to make you burn fat. It's going to make you lose weight, a lot of weight. So have you heard of these nitrophenol pills that started to getting used around the Second World War? Have you heard of that one? Good history class? So what happens is that during the Second World War, the men went to war, and there was nobody left to work at the factories, like bomb factories, right? So then the women got to work at the bomb factories. And they were using TNT and the nitrophenol and all this stuff. And they were getting very skinny. And then some other women are looking at these women and saying, wait a minute, you're getting skinny, I have to work out and lose weight, and you're getting skinny just by working at the factory? Not fair. So then everybody decided to take the nitrophenol and get skinny. And what's the problem with it? It also kills you. So yeah, you're gonna lose all the fat, like all your body fat and your body with it. It can cause hyperthermia and you're gonna die. So it's a bad idea to take the nitrophenol to lose weight. Although it would sound like a good idea at first. Okay? Why? It puts holes on the inner mitochondrial membrane. All right, so I'm going to stop it here. I know it's very dense, it's a lot of information here, but make sure you catch up with it.